they present, we're going to have the mic open for 3 questions. If there's any other questions at the end, we are more than happy to have them answered, but that's only if time permits. So think about questions while they are presenting. They will be answering any kind of content-specific questions. So without further ado, I'm just going to let it be student run. We have our 2 MCs. Okay, we have Mika and we have Claire. Please welcome them up. Nice round of applause.
The next common term you'll be hearing a lot about is CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, and it's a powerful gene editing tool which can alter the DNA of specific organisms. It allows precise changes that any other gene editing tool cannot do. And lastly, you'll be hearing a lot about regenerative medicine, which is also referred to as stem cell therapy. It's the process of repairing human cells, tissues, or organs that have been aged or diseased. It uses your body's own cells to promote healing. And advanced studies using regenerative medicine use biomaterials to create tissues and organs that can be implanted into your body. In this symposium, we'll have 10 total presenters. 2 from each class, starting with class 700 and class 704. So please give it up for our first presenter, Safa, from class 700. Thank you so much. My name is Safa Dua from class 700, and my focus question is how can CRISPR technology affect the prognosis of individuals with kidney disease? My thesis statement is CRISPR technology can affect the prognosis of individuals with kidney disease by providing targeted therapies that can cure genetic mutations,
reducing disease progression, and encouraging kidney regeneration. So before we start, here's some basic background knowledge regarding CRISPR. What is CRISPR? What does it stand for, and how does it work? CRISPR stands for Clustered Regularly Interspace Short Palindromic Repeats. First founded in 1987 by biochemist Jennifer Doudna and co-founder Emmanuel Charpentier, CRISPR is a gene editing tool that allows doctors and researchers to genetically modify DNA. How it works is RNA, which stands for ribonucleic acid, guides the Cas9 enzyme to a specific target site. The target site is the damaged gene. The Cas9 enzyme then acts like molecular scissors, cutting at the target site, allowing the gene's natural repair mechanisms to keep intact. There are two terms that appear in a few of my slides that I just want to clarify before we begin. CKD, which is just an abbreviation for chronic kidney disease, and the term renal, which is a synonym of kidney, but if you come up, I will make sure to clarify it. So what are the three effects of CRISPR technology on the prognosis of individuals with kidney disease? 1. CRISPR technology can provide targeted therapies that can cure genetic mutations. 2. CRISPR technology can reduce disease progression. And 3. CRISPR technology can encourage disease or kidney regeneration. This image right here shows the 5 different applications of CRISPR. Agriculture, drug discovery, industrial biotechnology, gene drives, and human therapeutics. Today I'll be mostly focusing on human therapeutics as that does make up most of my topic. Doctors and researchers can create and tailor specific personalized treatments based on a person's genetic makeup. This obviously varies from person to person as not everyone has the same DNA or genetic sequence. There are 3 traditional common forms of CRISPR in which most patients end up undergoing: drugs, direct injection, or immediate surgery. There are also 3 important factors that can impact what treatment a patient can end up undergoing: their age, health, and what their body needs. These personalized, perfectly tailored treatments for a patient's specific needs can help speed up recovery and reduce the need for other treatments like dialysis or kidney transplants. Most of us are familiar with the term kidney transplants, but not dialysis. Dialysis is a treatment in which 2 needles are inserted into a patient's arm. One suctions out blood, runs it through a machine that filters out waste, toxins, and excess fluids from the bloodstream, which then returns back into the patient's bloodstream through the other needle. Dialysis and kidney transplants are most commonly the last option, and not many people opt for it on the first treatment or trial because of low success and survival rate as well as cardiovascular complexities. According to a report from Shanghai University in Shanghai, China, patients' results improved by 24% post-procedure with limited side effects. 24% is an average of all 3 treatments, drugs, injection, and surgery. And 24% may not seem like a lot when I say it out loud, but when it comes to fatal diseases like chronic kidney disease, any improvement in the right direction shows a positive response to treatment and an improved prognosis. CRISPR technology can reverse pathogenic variants made to human podocytes. Pathogenic variants are changes, most commonly damages, made to a genetic sequence. Podocytes are essential cells that filter out our waste and toxins, as well as excess fluids from our blood. If our podocytes are damaged or dysfunctional, bacteria builds up, then causing diseases like CKD, as well as slowing kidney function. This image right here shows chronic kidney disease stages progressively getting worse, labeled eGFR. An eGFR stands for estimated glomerular filtration rate. It shows doctors and researchers how well your kidneys are at filtering out waste and toxins from your blood, specifically estimating how much blood can pass through your glomeruli each minute. The standard way to test eGFR is with a simple blood test that is then later analyzed to measure your creatinine levels. Only patients suspected of chronic kidney disease take this blood test. If your eGFR blood test comes out from 120 to a 90, your kidneys are perfectly functional, at the right pace, and filtering out waste and toxins like they should be. 90 to 60 earlier stages of kidney disease. Maybe your kidneys are a little slower, not filtering out waste and toxins normally. 60 to 15 is kidney disease, and 0 to 15 is kidney failure. So a few scholars from Harvard compared two EGFR blood tests, one before and one after CRISPR treatment. Between the two, blood tests showed a 43% improvement rate, which is very close to 50%, showing a positive response to treatment and an overall improved prognosis. CRISPR technology can stimulate and regenerate damaged kidney tissues. Damaged kidney tissues is a large percent or portion of your kidneys with cells that are damaged or dysfunctional. CRISPR technology, reversing damages made to your filtering cells that filter out waste and toxins from your blood, can enhance kidney function and improve kidney filtering capabilities as a whole, preventing transplants, dialysis, and slowing disease progression. Compared to other gene editing tools, such as TALEN, TAIL, and zinc finger, CRISPR's success rate is the highest. All different gene editing tools specialize in different things. So CRISPR technology may specialize in stem cell therapy, but zinc finger specializes in engineering nucleases, TALEN specializes in DNA binding, and TAIL is sort of a mixture of both. This graph shows CRISPR's success rate on kidney disease from 2004 to 2024. Although you see a small drop between 2020 to 2024, that's only because of COVID-19 and how many labs have closed during that time. In these patients, most, 60%, show a positive response to treatment and an improved prognosis, leading scientists and researchers to the conclusion that CRISPR has a 5 to 60% average success rate on all previous lab-ran tests and previous patients. So, in summary, CRISPR technology can affect the prognosis of individuals with kidney diseases by providing targeted therapies that can cure genetic mutations. Think back to the personalized, customized treatments that are perfectly tailored for patients' needs. Reduction of disease progression. Think back to the EGFR and the blood tests that help doctors and researchers get a better understanding of what patients need. Stimulating and encouraging kidney regeneration. Think back to the damaged tissues and filtering capabilities being enhanced. All of which provide for a more improved and effective prognosis for individuals suffering from CKD. These are my presentations. And
thank you, Sala. That was wonderful. Now we have time for 3 questions from the audience. Please make sure that they're all content specific.
So if someone has their kidneys like 5 to 10, is it like really bad?
If your EGFR blood test comes out from a 5 to 10, is your question? That's kidney failure. So yes, if a patient's EGFR blood test comes out from a 10 to 5, that's dangerous for their overall health.
Is there a cure if you get like kidney failure?
My focus is how CRISPR technology can help reverse damages, potentially healing kidney failure. But as of right now, I don't have an idea of other treatments, but I do know about CRISPR.
There's like normal, is there like above normal, like where like it's too high to like that?
That's a great question, but wasn't the ultimate focus of my research. Although if that's something that interests you, you have many opportunities to look deeper into it next year.
All right, that's it for questions. Thank you, Safa. Our next presenter is Chelsea from class 700.
Thank you for introducing. I'm Chelsea Gold from 700. Focus question is, how do the ethical considerations of de-extinction affect the progress in the scientific field? Here is the dodo bird, and above it is the Tasmanian tiger, also known as the thylacine, which I will refer back to later. My thesis statement is the ethical considerations of de-extinction affect the progress in the scientific field by compelling companies to respond to public's opinion, accommodate conservation concerns, and revive animals that serve an ecological function. Ethical considerations are principles and standards that guide decisions and actions to be morally right and fair, often in practice and research. Conservation is the protection, preservation, and restoration of wildlife. Now, de-extinction is the process of creating an organism that resembles an extinct species by recovering its core genes. There are 3 ways to go about it: backbreeding, also known as selective breeding, cloning, and gene editing. The picture here shows gene editing using the woolly mammoth as an example. So we take a fragmented DNA of a frozen mammoth and take what makes a mammoth a mammoth: larger tusks, more fat, adaptability to cold, and insert that into the DNA of an Asian elephant, the closest living relative. Now we have modified DNA. DNA is the set of instructions that tell your cells how to grow, reproduce, survive, and determines how your body looks like. With this modified DNA, a small electric shock, and a blank egg, we get a developing embryo. An embryo is the early development stage of any multicellular organism. That embryo would be in a serial mother or artificial womb, and the expected result is a mammoth-elephant hybrid. Because we are going to mention them a lot, what is Colossal Biosciences? Colossal Biosciences is a genetics and biosciences company founded in 2021 by Ben Lamb and George Church to focus on conservation and de-extinction. The World Animal Foundation predicts that 15 to 37% of all species will go extinct by 2050, an alarming rate that Colossal believes they can help prevent. They plan to bring back the dodo, the Tasmanian tiger, also known as the thylacine, as I have referred to in the beginning, the woolly mammoth, and the northern white rhinoceros. They plan to release their first de-extinction animal by 2028. We would still be in school by then, so it is important to consider what companies like Colossal are doing about our concerns with the phenomenon that's going to affect the world. For one, Ben Lamb, co-founder of Colossal Biosciences, wants people to have the complete picture, educating them. He says that they tend to agree in the end. They also allow cameras in to document things. For example, they are working with James Reed, director of the Netflix documentary My Octopus Teacher, on a 5-year-long documentary series. Ben Lamb also prefers criticism from someone who is informed so that they can take the best feedback and improve. However, why not? Why should we bring back the past when we could keep the present from becoming history too? Why not use these supplies on endangered species? Colossal says they are bringing back the past to create a better future. They are helping endangered species, including ones related to de-extinction targets. For example, elephants are related to woolly mammoths, and elephants suffer from elephant endotheliotrophic herpes virus, which causes a fatal hemorrhagic disease that involves excessive bleeding or bleeding disorders. 20% of baby elephants die from elephant endotheliotrophic herpes virus every year, some sources going as high as 85%. Colossal Biosciences can make the elephants more resistant to the virus by creating vaccines and editing the gene that makes them vulnerable to it. ReWild, an organization that restores and protects the wild, got feedback accusing Colossal Biosciences of stealing money from conservation. Colossal Biosciences refuted the claim, saying they are bringing innovations to these organizations. This picture here depicts some of their partners, which you can find on their website. They currently have 48 conservation partners. They also give away free technology that have an application to conservation, including gene editing and tissue engineering tools. Through Colossal Foundation, launched in October 2024, they can support these conservation partners. I'm going to use Vikash Tataya as an example. Tataya is a conservation director at Mauritian Wildlife Foundation, which you can find on this picture. He was skeptical of de-extinction at first, but a year later, he changed his mind, believing that the return of the dodo bird could simultaneously help life on Mauritius. Some of the reasons why he changed his mind was, instead of drawing away efforts on conservation, Colossal and Mauritian Wildlife Foundation's partnership led to more attention on Mauritius. Some of the things that Colossal has done for them is remove invasive species and restore habitats. Still, there are more cons than pros. This could end as badly as the Jurassic movies, with foolhardy scientists messing with gene editing. Companies choose animals that serve an ecological function. For example, George Church, co-founder of Colossal Biosciences, believes woolly mammoths are ecosystem engineers. The Arctic permafrost where they live stores a ginormous amount of carbon, which is part of carbon dioxide, a major greenhouse gas. Greenhouse gases trap heat on Earth, which causes climate change. Climate change is already a problem, so it is melting the Arctic permafrost, which then releases carbon and further feeds global warming. Woolly mammoths can trample woods into grasslands, which more effectively absorb carbon and less sunlight. This could reduce climate change and protect the Arctic permafrost. As for the dodo, the Tamala coke tree, also known as the dodo tree on Mauritius, has been struggling to reproduce since it went extinct centuries ago. The dodo could help disperse the seeds of the fruit by chewing and digesting it. Their digestive systems could break the tough outer coating of the seeds, allowing them to sprout. Finally, once the most common bird on Earth with a population of billions, the passenger pigeon took a key role in clearing woodland, thus rejuvenating them. This picture is by Revive and Restore, the organization spearheading this project. Revive and Restore believes that passenger pigeons can cause canopy disturbances, which then causes early successional stages, both which are key to completing the life cycles of many plants. Successive colonization is where different species of plants take over an area, creating a more complex plant community over time. This helps plants in various ways, some of which include creating more genetic diversity, adapting them to the changing environment, and dispersing seeds. All three of these animals are examples of what de-extinction can do for our planet. In conclusion, companies address ethical concerns by teaching and listening to the public, lending a hand in conservation, and de-extincting animals that contribute to the ecosystem in some way. As I have mentioned at the beginning, Colossal Biosciences plans to release their first de-extinction animal in 3 years. The passenger pigeon is expected to return in 7 years. Incredibly near, yet I had no knowledge of this before 7th grade. I did not even know the word de-extinction existed. It is a groundbreaking scientific achievement that stirs many concerns about what people with this kind of power in their hands would do. Are they playing God, meddling with nature? Is this a waste of time? How can we make sure that these hybrids benefit our planet and not bring harm? It's up to you and everyone else to ask the right questions and make the best decisions for a future when woolly mammoths or passenger pigeons roam the earth. Thank you. Yes, that's what they would do first before they release them to the wild.
Is it possible to de-extinct an animal even if there's no animal on Earth that's related to it?
It's probably best that we de-extinct an animal with something that's related to it, though it's not impossible to de-extinct something that isn't related to it.
Recently, scientists were able to bring back the dire wolf, which was an extinct species from 13,000 years ago. Do you know like the benefits of that?
That's a great question, but not the focus of my research. If you are interested in that, you could research that next year in 7th grade.
Thank you for that presentation. Now we're going to introduce our presenters from class 701, starting with Justin.
Thank you for the introduction. Hello, my name is Justin Cortez, class 701. And my focus question is, how can regenerative medicine affect individuals with athletic injuries? My thesis statement is, regenerative medicine can affect individuals with athletic injuries by providing safe treatment alternatives, amplifying the body's natural healing ability, and providing less risky treatments. So you may be wondering, what is regenerative medicine? Well, regenerative medicine uses cells which make up all living things, stem cells, which are cells that can turn into any cell in the body, and genetics, which are passed down traits, to regenerate damaged organs, tissue, and muscles. But by what I mean to regenerate is to regrow those damaged parts. Such treatments that are considered regenerative medicine are platelet-rich plasma, prolotherapy, stem cell therapy, and more treatments. So right here are the two treatments I'm going to be mostly talking about, which are platelet-rich plasma and prolotherapy. Platelet-rich plasma is similar to stem cell therapy, but instead of extracting stem cells, blood platelets are extracted from the patient. Blood platelets are small, colorless cell fragments that form clots and stop and prevent bleeding. A reason why blood platelets are extracted are due to their natural healing properties, which are growth factors. So after the blood platelets are extracted, they're put into a centrifuge to separate the different parts of the blood. So your blood has four parts. It has the plasma, which is the liquid form, the white blood cells, which deal with infections, and the red blood cells, which deliver oxygen, and the blood platelets, which I explained. So they're put into a centrifuge. A centrifuge is basically a machine that spins at high speed to separate the different components of the substance. It's basically the same force that a chair swing ride would do at an amusement park. So after the solution of platelet-rich plasma is created, it is injected into the injured area of the patients. So the success rate of this treatment is unclear because it depends on the injury. But even with that, the usage is expected to increase 66% by 2030. So over here is the whole process which I explained. The blood is extracted, put into a centrifuge, and it is platelet-rich plasma, platelet-poor plasma, which is the plasma with a low platelet count. The platelet-rich plasma is the plasma with a higher platelet. Then there is the red blood cells. The reason why they're like this is because, as you may have explained in science class, the substances with more density floats to the bottom, while the substances with lower densities float to the top. After the platelet-rich plasma is extracted and injected into the injured area. Prolotherapy is similar to platelet-rich plasma, as they are both injection treatments, but prolotherapy injects an irritant to cause your body's immune response. So what this does is that it increases inflammation. Inflammation is your body's response to an injury. It causes swelling and redness to the injured area. So this facilitates healing by attracting cells and increasing blood flow, which stimulates natural healing. This has a 80 to 90 success rate. So this picture shows it's a prolotherapy being injected to the injured area. This is injected into a knee. The benefit of both of these treatments is that they do not require any drug treatments and can be used with physical therapy. So in recent years, athletes have tried to avoid injury due to treatments being lengthy and unreliable. Such injuries that are common and severe include a tear of the anterior cruciate ligament, which is a ligament that stabilizes the knee, a tear of the rotator cuff, which is a group of joints in the shoulder, and muscle tears, which are structural damage to a muscle. So if an athlete ever gets these injuries, they would either retire or continue performing with restrictions. This can also be applied to the real world, as people who get these injuries would have restrictions on daily activities. As seen over here, these are the injuries I mentioned. This is the torn anterior cruciate ligaments. This is a torn muscle, the most common torn muscle, a torn hamstring, which is a group of muscles behind your thigh. Then there's a torn rotator cuff. So a torn ACL happens when there's a direct hit to the knee, you land awkwardly, or sudden jerky movement. A torn hamstring happens when your muscles stretch more than it's supposed to. Then a torn rotator cuff happens when the rotator cuff is worn down from repetitive activity. Athletes so far have been using bone grafts, which are treatments used to replace bone, painkillers, steroids, which are either performance enhancers or treatments used to treat inflammation, and there's more treatments. Drugs can cause health issues and they do not prevent joint deterioration, which comes from most injuries. The health issues that can be caused are gastritis, which is inflammation of the lining of the stomach, gastrointestinal bleeding, which is a disorder in the digestive tract where blood comes out of waste, and hematologic toxicity, which is adverse effects on blood flowing tissue. Bone grafts, on the other hand, they can be expensive. They cost a minimum of $2,000, and there's a chance of graft rejection. Graft rejection happens when your immune system detects the bone graft transplanted into you as foreign, meaning it detects it as a threat and starts attacking you. So you may be wondering, how can regenerative medicine help? Well, regenerative medicine is trusted by many people and doctors, as more and more doctors are being trained to handle these procedures, and they usually recommend this treatment if traditional treatments aren't as effective. So I have a question for the audience. So how many of you here know who Kobe Bryant and Tiger Woods are? So it seems most of you know who they are. So the reason why I say this is because both of these athletes have used regenerative medicine once in their careers. Kobe Bryant once used platelet plasma to treat his knee injury. Tiger Woods once used platelet plasma to treat his knee and back pain. Regenerative medicine can also amplify your body's natural healing ability and boost cartilage repair. So by what I mean to amplify is to make it stronger than it was before. This can help people who are aging, because as someone ages, they lose their abilities due to their body not being as capable as they were before. This can also help cartilage repair. Cartilage is connective tissue at the ends of bones that does not have a reliable blood supply. This means it cannot heal well naturally like bones do, so it needs help. This can be helpful for cartilage injuries or cartilage tears because they will need help on healing. According to Dr. Jasper Singh, clinical research results on regenerative medicine are generally positive on how it can potentially be a treatment for sports. This graph shows increase in the regenerative medicine market, showing about 190% increase from 2019 to last year. An increase from $13.3 billion to $38.7 billion. Currently, North America has the most usage, followed by Europe, Asia and the Pacific, and the rest of the world. In conclusion, regenerative medicine is a safe and trusted treatment alternative that can amplify the body's natural healing ability and heal cartilage. These treatments are also less risky compared to other treatments. This is my left slide. And my final slide. Thank you.
Thank you, Justin. Does anyone have any questions?
How much millimeters does the special syrup does it need to like heal a turtle?
Which one, prolotherapy or platelet plasma? Prolotherapy or platelet plasma? Platelet plasma? That's a great question. You should write it down on a paper and give it to your teacher, and I'll try to get in contact with you as soon as I can.
Any other questions? Right here.
Going back to the athletic injuries, which would you think would have the most long-term effect on a player?
I would think the tear of the anterior cruciate ligaments would last longer, as it depends on how much the tear is. If it's a complete tear, it could be like about 6 months to a year.
One last question.
If someone used steroids, what would be the side effect?
As I mentioned earlier, current treatments, steroid is a drug treatment, so it can possibly cause gastritis, gastrointestinal bleeding, or hematologic toxicity.
All right, so the next presenter is Andrew from 701.
Thank you for the introduction. My name is Ingrid Matthews from class 701, and my focus question is, how can Neuralink affect the methods physically disabled individuals use to complete daily tasks? My thesis statement is, Neuralink can positively affect the methods physically disabled individuals use to complete daily tasks by improving prosthetics, sight, and communication. Before I begin, I need you to know that my topic isn't directly stem cells, but it is related to stem cells because both stem cells and Neuralink can help treat ailments like paralysis, Parkinson's disease, and other types of brain injuries. Your brain works by sending information through your neurons to the other parts of your body. Neurons are nerve cells. Your body parts will receive the information from your brain, and they can send information back onto what is happening around them, allowing your body to work smoothly. Neuralink is a company that is making a brain chip that will allow you to interact with machines. I mean by interact with machines, you'll be able to think thoughts, and a machine will be able to carry out those thoughts how you intended it to. There are a couple designs that they're working to perfect, and these are some of them. So, Neuralink works by inserting hair-thin electrodes into the brain to record the neural signals that are being sent around. Inside the chip, there's a battery that lasts about 12 hours, and in order to charge it, you'll have to use this charger and hold it up to the battery, and it should charge manually. Over the years, more and more people are becoming disabled. 1 in 4 of today's 20-year-olds will become disabled before they reach the age of 67. One of the ways that Neuralink can improve disabled people's lives is by improving their prosthetics. For someone who is missing a limb, they will have to use a prosthetic. But prosthetics are very expensive since they usually have to be made custom for the person who uses it. They tend to range from about $20,000 to $80,000. Prosthetics using Neuralink could move with your thoughts. The chip would detect your brain signals and send that to the prosthetic, and the prosthetic could act out those commands how you wanted it to. The prosthetic could also send information to the chip, and using electricity, it could change your brain signals so your brain thinks it was feeling something that the prosthetic is actually feeling. This is a normal prosthetic leg. As you can see, it's not that complex and it's not nearly as good as an actual leg would be. And this is a Neuralink prosthetic hand that could be used as a normal hand would. For people who can't see or can't hear, there could be a camera or an audio recorder that could see or hear for them. It would send the signals of what it was seeing or hearing to the device, and then your brain would think it was actually seeing or hearing those things. Using this visual, it shows how it could simulate exactly how it would feel to see or hear those things. And so you would basically see or hear those as if you actually had working eyes or ears. Another way Neuralink could improve disabled people's lives is through communication. Some people are unlucky and can't speak, and they could have a speaker near them that could speak for them, because your brain would be sending around signals to try and get you to speak. But if you couldn't, well, you couldn't. But with a speaker and a chip, the speaker could speak for you. There are some problems with this, however, because why it's not out in the public yet, because in this visual you can see there's a battery and the microchips get to work, and the electrode threads that detect all the brain signals. Over the course of time, the electrode threads start to slip out of the brain and they can't detect the brain signals like they're supposed to. There is a way to fix this. You can have a brain surgery to get them implanted back into your brain, but lots of brain surgeries can be very dangerous for your health. My conclusion is that Neuralink can improve disabled individuals' lives by providing substitutes for lost limbs, lost sight, lost hearing, and communication. This is my work diagram.
Thank you for that presentation. Now we have time for 3 questions. Anyone from this side?
Because of Neuralink's high technology and how it includes communication and you have to insert it into your brain, wouldn't it be very expensive? I would just like to know the cost of it.
The estimated price range for Neuralink is about $10,000 to $50,000.
Where to like, what was it? The first thing was in the brain. What happens if you like don't have like any like internet, like no access to like technology? What do you do then? Is it like reliable?
The only way that the Neuralink chip really needs, like, the Neuralink chip is technology itself, but once you have that and the prosthetic, all you need really is the charger, but it would not be able to work unless it was charged.
Hi Ingrid, how are you? Quick question for you. How long has Neuralink been in development, and what is the biggest change that your research has seen during that time?
Neuralink has been in development for several years since they can't fully perfect it, and they have had trouble getting permission to do testing on actual humans. And the biggest change, I would say, is actually getting that permission because of the ethical considerations of the dangers of Neuralink and how it could, and how someone could possibly hack into your brain chip.
Thank you, Ingrid. The next presenter is a close friend of mine, so please give a warm welcome to Chloe Cheku.
Thank you for that introduction, Mika. Hello, my name is Chloe Cheku of class 702, and my focus question is, how can stem cells affect individuals with diabetes? My thesis statement is, stem cells can affect individuals with diabetes by reducing their glucose levels, restoring the function of pancreatic cells and the pancreas, and helping them produce their own insulin. Does anybody know someone who has diabetes? That's a lot of you. Would you like to share? Yes, I also have extended family who has diabetes and has to do that same treatment. But what is diabetes? Diabetes is one of the fastest growing chronic illnesses in the world, where in 1990, the population of individuals suffering with diabetes was 200 million, and it rose up to 830 million in 2022. There are two types of diabetes, and that's type 1 and type 2. With type 1, the body does not make enough insulin, and there's an 80% loss of pancreatic cells. With type 2, the body has trouble controlling blood sugar levels, and there's a decline in functional beta cells. Beta cells are cells that can produce insulin and are found in the pancreas. The pancreas, as shown in this image, is behind the stomach and is in the abdomen. How is diabetes treated? There are medications, which are injections of insulin, pills, one of them being metformin, and in this image, as you can see, the insulin injection and the metformin pills. However, scientists want to use stem cells, which are cells that can self-renew and differentiate into any type of cell in the body, to treat type 1 and type 2 diabetes. Stem cells can affect individuals with diabetes by reducing their glucose levels. With both types of diabetes, beta cells become dysfunctional, causing high glucose levels and damaged organs. To treat this, beta cells can be made in a laboratory to lower glucose levels and help reduce insulin. This image shows that in the normal blood glucose, there are fewer amounts of glucose in the blood, and in the high glucose levels, there are higher amounts of glucose, showing the difference between normal blood glucose and high blood glucose levels. Another treatment for type 1 diabetes is the VX-880, which is a stem cell-derived ISID therapy. ISID cells are cells that can produce insulin and are found in the pancreas. These cells are derived from a healthy donor and injected through the vein to the pancreas. This treatment was tested on a patient who has been suffering with diabetes for 40 years, and this patient had increased C-peptide, a decrease in insulin injections, and more controlled glucose levels. The company that developed the VX-880 was Vertex. Pancreatic cells and pancreatic structure can be restored by using mesenchymal stem cells, or MSCs. MSCs are multipotent stem cells, which are found in bone marrow. In a study, bone marrow and islet cells were combined and injected in mice with type 1 diabetes. These mice had increased activity in pancreatic cells, and this took place in 2021 in Stem Cells International. This shows the bone marrow cells, which are derived from the bone marrow, red bone marrow, and this is an up-close image of the bone marrow cells. In an additional study, MSCs were injected in mice with type 1 diabetes, and these mice had improved glucose levels, pancreatic function, and structure. This shows a difference between normal islet cells and type 1 diabetic islet cells. When the pancreas is damaged, there is risk for insulin resistance, which is when the body literally resists insulin. This can cause type 2 diabetes. In a study, mice had human adipose MSC injection. Human adipose cells are body fat cells, and this stopped the progression of cell death, which is also known as a loss of beta cells. This shows that this was done in high-fat mice in 2021. They used human adipose MSCs. This is the amount. They did it through the vein every 2 weeks up to 1 month, and this stopped progression of cell death, and this is who led the research, which was Shane and other researchers. Stem cells can treat people with type 1 diabetes by helping the body produce its own insulin. In a study in 2024 in China, dysfunctional beta cells were converted to functional ones in a patient, and this was done by having the fat tissue cells extracted and turned into induced pluripotent stem cells, which are iPSCs. iPSCs are cells that can self-renew into any type of cell in the body and turn into any of the cells. These iPSCs were converted to functional islet cells, and 1.5 million islets were injected in the patient's stomach. The patient, as previously mentioned, can produce their own insulin, and there is no need for insulin injections. As mentioned earlier, the VX-880 infusion is another treatment, and this is because the functional created cells are able to produce their own insulin. This shows the pancreas producing insulin, controlling glucose in the bloodstream. In conclusion, stem cells can reduce glucose levels, improve the function of pancreatic cells in the pancreas, and help people produce their own insulin. This can be done by using laboratory-created beta cells and the VX-880, inject MSCs into the body, and convert dysfunctional beta cells into functional ones. I hope those who have been affected with diabetes and know people who have diabetes learned about stem cells and how they can help people with diabetes.
Thank you, Chloe. Does anyone have any questions? Maybe you find out. Over here.
I'm correct. There's diabetes type 1 and diabetes type 2. Does this method work for both types?
Yes, but all the methods that were mentioned earlier can treat type 1 and type 2 diabetes.
Any other questions? What about type 3 and type 4 diabetes? Can it treat that?
This is not the focus of my topic, but that's a great question.
Okay.
And
What does metformin do?
metformin pills are a type of treatment, and this can help produce insulin, but this is before stem cells can treat type 1 and type 2 diabetes and potentially cure it.
Thank you, Chloe. The next presenter is
Thank you very much for the introduction. My name is Jeremy, of class 702, and I am here to talk about genetically modified mosquitoes. My focus question is how can using well-engineered mosquitoes affect the prevalence of mosquito-borne diseases? My thesis statement is that well-engineered mosquitoes affect the prevalence of mosquito-borne diseases by decreasing the female mosquito population by killing the genes that kills embryos and by making them disease resistant. I'm sure that almost every single one of you in this entire room are familiar with mosquitoes. Who here has been bitten by a mosquito? Raise their hands. Well, that's almost every single one of you in this entire room with their hands down. So I'm sure many of you who raised their hands will find what I have to say about this presentation interesting. This presentation is important because by listening to it, you can actually understand the hidden destruction that mosquitoes may cause simply by biting another person. So mosquitoes are the deadliest animal in all of planet Earth. They have killed over 52 billion people in all of recorded human history. And to put that into perspective, only about 108 billion people have ever lived, which means that almost half of all humans that have ever lived on Earth have died from mosquito-borne disease of some sort. Nowadays, in modern developed nations, like in the United States, where the healthcare is more advanced, and as conditions that aren't as ideal for mosquitoes, they aren't as much of a threat anymore. But in less developed nations, especially in Africa, where the healthcare is less advanced and it has conditions more ideal for mosquitoes, they are more common and still cause a lot of harm and suffering there. However, there's a new front in the war against mosquito-borne diseases, which is called the genetically modified mosquito. And over here, you can see a picture of the average everyday mosquito. So the first issue I'll be discussing involves an invasive species of mosquito called the Induced ensign. Invasive species is a species that is not native to the local ecosystem, and it causes a lot of harm to the local wildlife and potentially even the people living there. So ideally, you want to remove invasive species from the local ecosystem as fast as possible. But how can you do that? Well, a British company that goes by the name of Oxitec has developed a solution to do just that. So what is Oxitec's solution? Well, Oxitec's solution involves genetically modified male Anopheles mosquitoes. And what these male mosquitoes do is that any female offspring that the male mosquito may have will be killed off. Now you may be wondering, why would they specifically choose to kill off females? Well, female mosquitoes are the only kind of mosquito that actually drink blood and they spread disease. So by decreasing the amount of female mosquitoes in the population, they're actually decreasing the amount of potential bites and disease cases that can happen. And there will also be no more females to reproduce with, which eventually means that the invasive mosquito species will decrease in due time. So the plan is to release a group of these modified male mosquitoes with the gene into an area that's affected by this invasive species. So that way, the male mosquitoes can mate with the local wild females in that area. So that way, when the females lay the eggs of that modified male, all of the female eggs that will turn into a female will die off. But here's a very important fact to note: all male mosquitoes will survive until adulthood untouched. So that way, when the male mosquito grows old enough, is old and mature enough, that male offspring and then mate with other local wild females in that area, and then the process can be continued over and over again until eventually the invasive mosquito population will decrease or will be completely eradicated from the local area. Now you may be wondering to yourself, is this technology even safe to begin with? Well, there was survey by both the FDA, which stands for the Food and Drug Administration, and the EPA, which stands for the Environmental Protection Agency, and they both declared that these mosquitoes pose no threat to the local ecosystem or people living there. They're non-toxic and do not use any sort of chemicals in the process. They only target its designated species, which is the Anopheles ensign, and will go after any other mosquitoes that are native to the local ecosystem. And you may also be wondering to yourself, was this technology used before? Well, over in Brazil, they had a very similar problem of another species of invasive mosquito, And when Oxitec uses the exact same technology in Brazil, it decreased the invasive mosquito populations by up to 95%, which means that almost all of the mosquitoes, the invasive mosquitoes in that area, was eradicated. Now the next issue I'll be discussing involves another invasive species of mosquito called the Induced penside. Now, like I said before, we want to remove invasive species from the local area as fast as possible. But how can we do that? Well, scientists at the California Institute of Technology have developed a gene set called maternal affect dominant embryo arrest, or MADIA for short. And what this gene set does is, when it's implemented into a female mosquito, all potential offspring that the female mosquito may have will have the gene called MYD88 silenced in them. And what an MYD88 stands for myelodifferentiation primary response 88. And this gene is essential to the development of the embryo. So if you remove this gene from the development process, it can't even hatch and become a full-grown mosquito, which will overall decrease the general invasive mosquito population, which will be very beneficial for the local ecosystem. Now the last issue I'll be discussing involves a disease called malaria. I will be using this term, so it's best if you know what it is. So malaria is a very deadly mosquito-borne disease that can trace its origins back to ancient times and has killed billions of people in recorded history. Malaria is caused by a parasite called Plasmodium. Like many other mosquito-borne diseases, malaria is more common in wet tropical areas where mosquitoes are more easy to thrive. And symptoms of malaria include fever, muscle and joint pain, chills, and rapid heart rate. So what is a solution to counter malaria? Well, the solution is to simply go back to the source to prevent the problem from even happening in the first place. But first, in order to understand the solution, you first need to know how malaria can spread through mosquitoes. So first, a female mosquito bites a person already previously infected with malaria, and from there, the parasite that causes malaria, which is Plasmodium, and then enters the mosquito's body, and it goes into the midgut section of the mosquito. And from the midgut section of the mosquito, it could then make its way up into the salivary glands of the mosquito, where the glands where the saliva is produced. And it stays in the salivary glands until eventually the female mosquito bites another victim, and the Plasmodium can then enter through the saliva. And this cycle can start all over again with another healthy mosquito. When another healthy mosquito bites the person just infected with malaria. Dr. George Mopos at Johns Hopkins University has genetically modified mosquitoes to have boosted midgut immunity, which is important because, like I said earlier, in order for the Plasmodium to get into the salivary glands of the mosquito, it first needs to make its way past the midgut section. So by enhancing the immunity in that specific region, the malaria can't even make it past into the salivary glands in the first place. And this genetic modification proved successful with successfully suppressing the malaria parasites as well as other bacterial infections. But there's actually an unexpected side effect that came with this genetic modification. It caused microbial changes in the mosquito's reproductive system. And a microbial change is a change of the bacteria in a certain area in order to adapt to environmental changes. And what this microbial change did is that actually made it so that wild females preferred modified males, and wild males preferred to mate with modified females, which helped the genes spread out more effectively. And over here you can see the anatomy of a mosquito, and here's the midgut section and the salivary glands. You could see a scan of the Plasmodium parasite invading the mosquito's midgut area. So here's an example of this. Now, to test the animal's breeding behaviors, scientists mixed an even amount of modified mosquitoes with an even amount of wild mosquitoes in an enclosed environment to see how to make sure that the gene could spread around the population properly. And the scientists only expected about 75% of the mosquitoes to have the genes after a few generations, but in the first generation, it was actually over 90%, which means that in the first generation, almost every mosquito had the gene. It remained at 90% for 10 more generations. And even when you mix 10% modified mosquitoes with 90% wild mosquitoes, the genes still persisted after 3 generations, which shows just how effective this technology is. So over here, I've made some pie charts which details the initial populations of the mosquito, which is 50-50 down the middle, and the populations of the next generation, which is 90% compared to the 10%. In conclusion, the way that prevalence of mosquito-borne diseases can be affected is by releasing males with a gene set that kills off all of their female offspring, which therefore decreases the potential disease cases. By coding a gene set that, when implemented into a female mosquito, will kill off all of its potential offspring, therefore decreasing the overall invasive mosquito population, and by making them resistant to disease, which prevents them from even being infected in the first place. Thank you for listening.
Thank you for that presentation. Any questions?
So, you say that you want to kill off all of the female mosquitoes, right?
That's correct.
So, wouldn't that decrease the population of all the mosquitoes? And like, considering that mosquitoes are like at the bottom of the food chain, they technically feed like all of the other animals. So, wouldn't that be like bad?
Well, like I said earlier, it only goes after invasive species, which is the Regenerative enzyme. So invasive species is not beneficial and harms the local ecosystem. So it will only go after associated species and won't, and any species, mosquito species that is local to the native ecosystem will still remain safe and untouched.
If they kill out the female mosquitoes, how would they make the male mosquitoes?
Well, with all the female mosquitoes gone and with no more mosquitoes to reproduce with, all of the remaining male mosquitoes will invasive. Remember, these male mosquitoes are still invasive to the population, so eventually the modified mosquitoes will eventually die off, and the invasive mosquito population will eventually be eradicated from the local area.
Hi, I may have missed this in your presentation. How do they modify the mosquitoes? Do you know the process in which they use like CRISPR or anything like that?
Well, unfortunately, when I was researching, it never really said what kind of technology they use to genetically modify the mosquitoes, but that's a really good question, and I could potentially get back to you.
Thank you so much, I appreciate that.
You're welcome.
Good morning, everyone. My name is Zizin Chen from class 703, and my focus question is, how do fetal stem cells compare to embryonic stem cells when used in research facilities? My thesis statement is, fetal stem cells and embryonic stem cells show similarities in public opinions, treatment results, and advantages. Right now, you might be asking, why should I care? What does this have to do with me? Well, stem cell research like the ones I will be talking about today can greatly impact the future of medical advancements and life-saving treatments. Although this might sound unsettling to some, but the truth is there will be a great chance that you or somebody that you love will be affected by a serious disease. Taking cancer as an example, 40 to 50% of people are diagnosed with cancer at some point in their lives, which is roughly around 800 students that come from McKinley who have the potential to develop cancer. That is a great percentage with just cancer alone. Now, I would like you to imagine someone that you care about facing a disease that can be treated and even potentially cured by stem cell treatment. What do you want to know what these stem cells are and how they can help? Well, today I'll be here to explain to you the functions of fetal stem cells and embryonic stem cells, their similarities and ethical debates, treatment success and risks, and in general, their roles in research. At this point, there should be another question popping up in your head asking, what are fetal stem cells and embryonic stem cells? Fetal stem cells are harvested from a fetus at 7 to 12 weeks of development. At this stage, the fetus is starting to develop organs like the brain, the heart, and the liver, which just shows that fetal stem cells are starting off to be a bit more differentiated. A fetus is the offspring of a human or an animal in the womb after the stage of an embryo. However, embryonic stem cells are from a 5-cell blastocyst, which is just an early stage of an embryo, from a female egg cell fertilized by male sperm cells in a laboratory. Right now, I would like to place a disclaimer that fetal stem cells are a really controversial topic. Not many people like to discuss them. That's why some of the information you see on my slideshow might be a bit more outdated. Both types of stem cells face ethical concerns affecting research policies. One of the biggest debates when it comes to using these stem cells for research is the way they are harvested and the way they are obtained. Embryonic stem cells require the destruction of an embryo, which to many people they find disturbing because they see embryos as the start of life. And destroying that start of life just for scientific research may seem cruel. And this ethical concern can also be tied to religious and personal beliefs. Similarly, fetal stem cells are obtained from aborted fetuses, which again, many people are against abortions and debate on whether or not it is appropriate to use tissues from terminated pregnancies. These ethical debates influence policies like laws, funding, and research limitations. Institutes and research facilities like the NIH has to follow these strict ethical guidelines affecting lab work. Right here, I have a poll from 2024 from News Gallup on whether or not these stem cells should be used for research. What I find really interesting about these polls are there's a great difference in the percentage of people against this idea from year to year. For example, there'll be a high percentage of people against this idea. The next year, this percentage will decrease by a significant amount. The year after that, this percentage will go up again. But the most recent poll that we have as for now is the 2024 poll, where 63% of people found this using these stem cells acceptable, 33% were against it, and 4% was still unsure. To address these ethical controversies, research facilities and scientists have been looking out for alternative stem cells, and these stem cells include adult stem cells and induced pluripotent stem cells. Adult stem cells still have the ability to become different cells in the body, but are just more limited to and more restricted to what it can become. Induced pluripotent stem cells are basically just adult stem cells being reprogrammed by scientists to act like embryonic stem cells, giving them more freedom and a bigger range to what it can become. Induced pluripotent stem cells carry the patient's own DNA, reducing the risks of rejection. Genetic rejection is when something that does not belong in your body gets into your body and your immune system is trying to kick it out. Now, this process can cause some negative effects, such as inflammation, infections, and blood clots. So it is a good idea to use alternative stem cells that can avoid this danger. Researchers also prefer adult stem cells for its safety and predictability, since fetal stem cells are known for its dangers of unpredictability, which I'll be advancing more on in my slides later on. Over here, I have a diagram showing how induced pluripotent stem cells work. Somatic stem cells, which is a more formal way to say adult stem cells, taken from the patient's body, reprogrammed into induced pluripotent stem cells, and now has a bigger range to what it can become. Research facilities study fetal and embryonic stem cells to learn about the potential and risks in research. Although there have been many controversies surrounding these stem cells, there have been shown successful treatment cases, such as in a case study, fetal stem cells were able to cure a child with bone marrow failure. The child was waiting on a bone marrow transplant, but at the time there were no suitable donors available. The child was then treated with 2 rounds of fetal liver cells, ultimately treating him. In another case study, embryonic stem cells were able to help treat 2 people with age-related macular degeneration. Macular degeneration is a disease that affects a person's vision and a person's eyesight. These studies from research facilities highlight their potential in medical treatments. Over here, I have an image showing the comparison between a normal eye and an eye that has been affected with macular degeneration. As you can tell, the spot over here is a lot darker than the spot on the normal eye. However, these treatments do come with risks. Fetal and embryonic stem cells have been shown to cause teratomas, which are non-cancerous tumors, or just abnormal growths in general. Fetal stem cells have also shown inconsistent treatment results when treating Parkinson's disease, showing little to no improvements in older patients and even worsening the conditions in younger patients. These risks increase the need for research facilities to do further research on these stem cell therapies, ensuring their safety and effectiveness. For instance, facilities like Stanford University conduct preclinical research to prove its safety before actual human trials. Both fetal and embryonic stem cells play an important role in research advancing the future of medicine. Both fetal and embryonic stem cells can provide unique benefits in research facilities, advancing research when researching different diseases. Fetal stem cells, as I have stated before, contains multiple organ-specific cells, helping researchers develop targeted treatments. Embryonic stem cells can also be used for regenerative medicine, which, if you are on an organ transplant waiting list, you'll have to wait until a suitable donor is available, and that can take a long time. Using embryonic stem cells for regenerative medicine can hopefully act as an alternative, reducing the wait. Research facilities and institutes like Harvard Stem Cell Institute use them to model diseases such as Alzheimer's and Parkinson's disease, explore organ regeneration, like I have stated here, test new treatments, which all ties to advancing the medical process. Over here, I have a diagram showing how regenerative medicine works, starting from RNA therapy, stem cell therapy, organoids, gene therapy, all the way to cell transplantation. To wrap this all up, here are my key takeaways. Ethical debates influence policies. Both fetal and embryonic stem cells show treatment potential but poses risks. They advance research in research facilities. These factors influence the future of medicine, making stem cell treatment an important field to understand. Here is my work.
Keeping all the risks in mind, is the treatment 100% going to work, or is there chances that it won't work?
With these risks in mind, fetal and embryonic stem cell treatment can, of course, have risks. They won't 100% work. There will be risks that you have to watch out for, and it won't always work. So yes, exactly.
Hi Zixin, how are you? I'm back. What is something that excites you most about your research?
Something that excites me the most with my research is to learn about the abilities of fetal and embryonic stem cells and their potential when actually being able to treat people and help people with their diseases. Yes.
Thank you so much. You're doing great.
One last question.
If you had another chance to make another slideshow like this, would you take the chance?
I love this question, but this is a more experience, and double AM question. Is there any other content specific questions that I can answer? Thank you for that question, though.
Any questions?
Oh.
Thank you, Zishan.
That was a great question. We're going to try to, any experiential questions that you want to ask at the end, if we have a panel, if we have time for that, you can ask then. We're just trying to ask content-specific questions at this moment.
Thank you for that. Our next presenter is Cynthia from class 703.
Thank you, Claire, for introducing me. Hello, everybody. My name is Cynthia Liu of class 703, and my focus question is, how do induced pluripotent stem cells compare to embryonic stem cells in research facilities? My thesis statement is, induced pluripotent stem cells and embryonic stem cells differ in their quality and purpose with their shared gene expression patterns. So first, we need to understand what are induced pluripotent stem cells? Well, induced pluripotent stem cells are adult cells that have been genetically reprogrammed to become any cell in the body. Genetically reprogrammed is when genes within a cell is either repressed or activated in order to convert one cell type to another. So as you can see here, it shows us that the adult fibroblast cell, a fibroblast is a type of cell that contributes to the formation of connective tissue. It goes through reprogramming factors, and then it can turn into all these type of cells. I will also briefly discuss what embryonic stem cells are. Embryonic stem cells, like said before by my peer Ziza and Chen, are derived from embryos, specifically blastocysts, which are 3 to 5 days old embryos. So as you can see in this picture, it depicts the embryonic stem cells being derived and the culturing in a laboratory. And then using different culture conditions, you are able to turn these embryonic stem cells to nerve cells, muscle cells, gut cells, and even whole organs. We also need to understand what epigenetics are. Well, epigenetics is the study of how your behaviors and environment can cause changes to how your genes work without changing the sequence itself. So what this means is that it changes how your body reacts, how your body reads the DNA sequence without actually changing it. So as you can see here, this diagram shows how a woman who smokes can cause her daughter and her daughter's reproductive cells to have an abnormal increase in hunger hormone and a higher potential risk of obesity. So as you can see in this picture, from generation 1, it tells us she's smoking. And as the generations progresses, you can tell that she has a higher risk of obesity as her body grows larger. We also need to understand what a gene expression is. So a gene expression is a process by which the instructions in a cell are used to create a functional product. For example, a protein that the cell needs to function. So as you can see here, the DNA transcripts into mRNA. RNA is mainly used to translate genetic information into something that the body understands. So when translation happens, the body will understand this and create a protein. So induced pluripotent stem cells have a difference in their quality. For example, they are highly accessible to many groups, which of those 80 groups, there are scientific research companies and private organizations, and etc. The variability can limit use because there are speculations that these cells are potentially tumorous and cancerous. There are also hundreds of cell lines that are spread across globally, and epigenetic will not influence these cells because studies shown during the reprogramming phase, any signs of epigenetic marks disappears. So as you can see here, this is another diagram of how adult cells are being reprogrammed. So when they are recombinant, they can turn into the brain, the heart, and even the lungs. And embryonic stem cells are also different in quality. Like my peer Z Zen mentioned before, they have very limited research because of how controversial they are. It is considered morally wrong to destroy the blastocyst because it's considered to kill the life inside. There is also less concerns of these cells because there's a higher understanding of where these cells are sourced from, and advancements in technologies, there has been more trust in regulations as time goes by. And epigenetic will influence these cells because these cells are derived from an embryo, and an embryo is a fusion of the fertilized egg, which is the mother's DNA, and the father's sperm. So as you can see here, it shows us a diagram of how this works. So the sperm and egg joins, and then the embryo develops for 5 days to 7 days, and then the blastocyst is developed, and then they remove the inner cell mass, which contains the embryonic stem cells. And when they grow it into a dish, it can turn into many different types of specialized cells. Induced pluripotent stem cells also differ in their purpose. For example, induced pluripotent stem cells reduces risks of immune rejection due to their cell properties. So immune rejection is when the body detects a foreign object and attacks. This happens mainly with organs, so about 10 to 15 organ recipients experience this. And they are also more useful for experiments. For example, this diagram shows that when they are differentiated into specialized cells, it can be used for disease modeling, drug screening and discovery, and also toxicity tests. Both the drug screening and toxicity tests can help create new drugs that will all circulate back to the patients. Embryonic stem cells also differ in their purpose. Compared to induced pluripotent stem cells, embryonic stem cells are preferred in organ development. Because, like I mentioned before, induced pluripotent stem cells have speculation that they are potentially tumorous, it is led to embryonic stem cells being the first type of stem cell to be used in the developing organs. They are also applied as a tool for studying genetic diseases. For example, fertility doctors from Chicago were able to make embryonic cell lines from an embryo that was rejected due to a genetic defect. They were able to create cellular models for degenerative diseases like Huntington's disease and Marfan syndromes. Huntington's disease is a disease where nerve cells break down over time, and Marfan syndrome affects connective tissue, making it visually weaker. And this pie chart shows us that around 75% of the medical research utilizes human-induced pluripotent stem cells over human embryonic stem cells. And this chart was created by me through statistical data gathered through my research. Not only do they differ in their purpose and quality, they also differ in their gene expressions. So induced pluripotent stem cells have increased variability and instability, which means that because of this, it will affect the product of the sequence. By affecting the product of the sequence, it will cause mutations and also mistakenly copy variants. There are also potential risks of diseases in their genes. For example, because they're taken from a person's lifetime, it shows all the possible diseases in their genes. So if a person smokes, it will show that they have potentially have lung cancer in their genes. Embryonic stem cells also differ in their gene expression. Compared to induced pluripotent stem cells, they have stable and well-defined patterns that are consistent with self-renewal and pluripotency. They are also not influenced by physical changes. Because these embryonic stem cells are derived from an embryo, which is a fusion of both the father's sperm and the mother's DNA. And they also share gene expression patterns. For example, they have large defining modules and small evolutionary differences. And you may be wondering, why is this important? Why does this matter to me? Well, first of all, we cannot live without stem cells. For example, heart diseases causes 1 in 5 deaths in the nation, and that is a lot. So now imagine yourself being told that your heart was failing, and then now imagine yourself Now imagine yourself being told that your doctor are able to take your skin cells, turn it into heart cells that was made from you, and you receive a new heart, which is just like your old heart. And there's no rejection, and there's no donor necessary. And this doesn't only apply to organs. Sooner or later, you are able to get and use medicine that was tailored just for you. And all of this means a better medical aid and more potential to recover from life-threatening diseases. This makes living more accessible for everyone. So for example, enjoying a walk through the park may mean the world to someone who is diagnosed with fatal diseases. With stem cells, induced pluripotent stem cells, and embryonic stem cells, this is possible. This is my mention essay, and this is our Friday.
Thank you for that, Sophia. Now we're going to be taking questions. None?
So, like in the example that you said, like when you're getting a new heart, does it have to be a person that's like related to you or have similar DNA?
Yes. When you receive a new heart, it usually has to be able to fit your organs and correlate with them, and usually you do not need to have similar DNA because everyone has different DNA. So if the organ fits you, then you are allowed. Then you can have it as your new heart.
So if your heart fails, will you be able to get a new heart in time?
What do you mean? Like by getting a new heart in time?
Will you able to heal it in time?
If the heart,
With the stem cells.
I actually don't know a lot about that topic because I did research about it. But usually, when the heart is failing, you can save it with stem cells. Like I use stem cell therapy and other things that are designated to help the heart.
If the heart is failing, like if the half of the heart is failing, can the stem cells like repair it?
Yes. Stem cells are also used in regenerative medicine. So by using stem cells, you are able to create new tissue that will potentially help save the heart.
Okay, thank you, Cynthia. Our next presenter is Yushin Wee from class 704.
Thank you for the introduction. Hi, my name is Susan Lee from class 704. My focus question is, how can induced pluripotent stem cells affect a person suffering from spinal cord injury? My thesis statement is, induced pluripotent stem cells can affect a person suffering from a spinal cord injury by regenerating damaged neurons, eliminating pain, and encountering risk of tumorigenesis. First off, why should you care about this presentation? Well, induced pluripotent stem cells have great potential in cell therapy. However, not many scientists or researchers actually use them for cell therapy. Cell therapy is a field aiming to use stem cells to restore damaged tissues. Now we have to understand what spinal cord injury is. Spinal cord injury is a damage in the spine that may be occurred through motor vehicle incidents, diseases in the spine, and a serious falls. These factors contribute to weaken the spine and cause a loss of function in the body. This brings us to the levels of the injury. Most severe being cervical, then thoracic, then lumbar, and least severe being sacrum. This image shows the 4 levels are categorized into the type of nerves that can be damaged. For cervical nerves, the head, hand, neck, etc. nerves are damaged. Over here, it shows why it's more severe because they control a larger portion of the body and it's closest to the brain. Then we have thoracic nerves. It damages the chest or abdominal muscles. This helps maintain a good posture in your body. Then we have lumbar nerves. It damages the leg muscles, which it helps support weight. And then we have sacral nerves. It damages the bladder and sexual function. These help get rid of waste in the blood. Now we have a data analysis on which level occurs the most. Around 60 to 75% occur in the neck area, which is the cervical level. The most severe level, it damages the head, hand, or neck. And around 20% occur in the upper back or chest area, which can be the thoracic nerve that damages the chest and abdominal muscles. 5 to 20% occur in the lower back, which can be either sacrum or lumbar. Introducing induced pluripotent stem cells. Induced pluripotent stem cells are cells that are reprogrammed from somatic stem cells, and they can become any cell in the body because they are undifferentiated. However, they may contain risk of tumorigenesis. Tumorigenesis is the formation of tumor, and tumor is an abnormal mass of tissue in the body when cells divide more than normal. In this image, it shows induced pluripotent stem cell colonies. These colonies are just induced pluripotent stem cells in a cluster. They can be used as regenerative medicine. Risk of tumorigenesis. So by being reprogrammed from somatic stem cells to induced pluripotent stem cells, it introduces genomic instability. An example of genomic instability would be chromosome aberration. Chromosome aberration is an extra or missing chromosome. In this case, for tumor, it's an extra chromosome. So the problem with an extra chromosome is that it causes genetic disorders. A disorder would be tumor, however, it could also cause other genetic disorders. Having tumor can cause pain because it spreads around the body, meaning it causes pressure in the body's tissue. Regenerating neurons and eliminating pain. So neurons are nerve cells, which are the units of the nervous system that send signals to the body when dangers are near. Here shows a process of induced pluripotent stem cells being taken from an adult human body, which is somatic stem cells, then it gets reprogrammed to become induced pluripotent stem cells. So once the reprogramming is over, it starts to target the neuron by differentiating into neural cells like neurons, and by doing this, it enhances regrowth of the axon. The axon is the long thread part that connects to the neuron that transmits the signal from the spinal cord to the brain. In conclusion, induced pluripotent stem cells can regenerate neurons by targeting the neurons by differentiating into neural cells. It eliminates pain by enhancing regrowth and enhancing axonal regeneration. And it causes tumorigenesis because it introduces genomic instability, it leads to chromosome aberration, and causes pain. This is my work cited on the list.
Are the stem cells injected directly into the spine?
Yes, it is. So once it injects, it targets the neurons.
Why is the cervical area the most damaged area?
It's the most damaged area because it is a more severe level since the head and neck or hand controls a larger portion of the body and is closest to the brain.
Can you specify which diseases?
That's a great question. However, that wasn't the focus of my research. If that's something you're interested in, it could be something you look forward to next year.
Thank you. And now it's time for our last presenter, Jasmine from 704.
focus question is, how can microgravity affect stem cells? Microgravity can positively affect stem cells by improving differentiation potential, revealing molecular changes that will not be seen on Earth, and increasing cell division. Stem cells are cells that are able to differentiate or develop into many different types of cells in the body. The picture on the right shows different types of cells stem cells could develop into. For example, stem cells could develop into red blood cells or fat cells. A microgravity environment is when gravity seems to be very small or weak. Gravity on Earth is referred as 1g, so microgravity would be 1/9 of the force of gravity at Earth's surface. As stem cells are cultivated in microgravity, storages like this are designed to prevent contamination, temperature changes, and other environmental factors that can impact the cells. Currently, there's a lack of patient-specific cells on Earth. Microgravity facilitates the differentiation potential of stem cells by enhancing osteoblast differentiation and strengthening. Microgravity also induces the cells to expand and differentiate. Stem cells cultivated in microgravity have shown molecular changes in different behavior. Microgravity further enhances regenerative capabilities of the cells as well as improves the body's immune response to illnesses. Regenerative capabilities refers to the cell's ability to repair, regenerate, or replace damaged structures. Microgravity increases cell division by increasing cellular aggregation. The properties in microgravity provides a more natural 3D state which helps mass produce the cells. A study by Marshall University has shown a larger aggregate of approximately 7 millimeters, which compared to a standard aggregate of 1 to 2 millimeters is much larger in size. This graph shows increased cell division when stem cells are cultivated in microgravity. 1g refers to the gravity on Earth, and SPC refers to microgravity or space. At 6 hours, space-grown stem cells were 120% more than those grown on Earth, and at 15 hours, 85% more. At 30 hours, 33% more. In microgravity, 81% of the neural stem cells were large. Larger stem cells result in more growth. A recent study by Spinal Institute of Neuroscience and Human Behavior has shown a twofold increase of the number of neural stem cells in microgravity. These images illustrates how microgravity advances and benefits society. Microgravity improves regenerative medicine, drug screening, and disease models on Earth. The picture on the right shows how scientists send stem cells to space for their altered differentiation potential, discovered molecular changes, and different behavior for possible clinical applications on Earth. So why are stem cells in microgravity so important? Microgravity provides a unique environment to uncover things we cannot observe on Earth. These insights could lead to breakthroughs in medical treatment as well as increased innovation for healthcare on Earth. The knowledge gained hold the potential to advance the future of stem cell treatment.
Thank you. Does anyone have any questions for her?
In the contaminant photo, what would happen if the cells were released? Would it affect the environment in any way?
You're saying if they're released from the storage? So the environment around it could, the contamination or temperature changes could alter their differentiation or growth in microgravity.
What are these tested on anyone? Like any person, like maybe like an animal?
That's a great question, but that wasn't the focus of my research, so you could research that next year.
There's one over here from a different class. Hold on.
So is there any ways that microgravity can negatively impact the stem cells?
Again, that's a great question, but that wasn't the focus of my research. I was investigating the positive effects of microgravity on stem cells.
I'm not sure if you defined this or not, but what is an osteoblast?
An osteoblast is a cell that forms and creates new bone.
Thank you. So, in a moment, we are going to have some speakers come up and answer some questions that were submitted by the sixth graders. But the audience can take a moment to stretch and take a rest. Please do not get out of your seats.
1 minute stretching, get it out, and then we're gonna have some answers. All right, great. I think we got some of that energy out. 8:16. Take a minute, sit down, please. So there have been many experiential questions that I know you want to ask, and one of the questions were asked at one point, and they were going to only answer content-specific questions. We have some questions that were submitted, so I'm going to give this back to Mika, and she's going to explain to you what's about to occur now.
Alright, so the questions that were submitted by the sixth graders are about IIM, the class IIM. And please keep your applause till every presenter gets a chance to come up and speak. The first person is Evan Chen from
Thank you for the introduction. My name is Evan Chen from class 700, and although I didn't present, my focus question was, how does regenerating joint cartilage with stem cells affect the transplant recipient? The first question is, what was your experience when you were finding sources for your topic? By Lucas E. from class 600. Finding sources for my topic was not too difficult. I mainly used the school database, which had multiple articles and magazines related to my focus question. We were also allowed to use a variety of reliable websites like the National Institute of Health, which covers a lot of different subjects. Overall, there are many sources of information that you can use, which makes researching about your topic easier. However, I do know that some of my classmates did struggle finding information, so depending on your focus question and your thesis statement, you may find different amounts of information. The second question, what are some things you wish you knew before you did IIM? Is it difficult for you? By Tala Ahmed from class 600. Entering seventh grade IIM, I did not expect the amount of work I had to complete. This year, I had to find magazines, videos, newspapers, and articles for both my slides and my essay. Having to use so many sources was difficult. However, the assignments were structured in a way that made this process easier. For example, for one assignment, I might be required to find no facts just using a video. And for another assignment, I might find no facts just using a website. Although the class is challenging, the guidance provided ensures that the work is not too overwhelming.
close friend of mine. Please welcome Huron.
Thank you, Mika, for that lovely introduction. And hello, my name is Yuran Liu from class 701, and I'll be answering 2 questions from class 601. First, I'd like to start off with saying my focus question, which was how can organoids affect the cancer research industry? The first question I'll be answering is submitted by Sofia San Miguel from class 601. Was this a cool experience, and what was it like? Do you have any advice for us when we go to 7th grade IIM? It really was a cool experience because we were able to research more about medical aspects such as stem cells and genetics. Advice I would give for when you start 7th grade IIM is that when you start your research or when you find evidence to support your thesis, you should try to research more in depth to better support what you're trying to explain through your projects. Other advice I would give is that during 7th grade, you should not procrastinate or overthink about how you would do. Overthinking could lead you to stress about your project and could worsen how you do. The second question I'll be answering is by Ezekiel Cruz from class 601. Were there challenges while you were researching? Yes, personally there were many challenges I had. The most of the challenges I faced was when I was researching. It was one of my challenges because some of the data I'd already researched did not support what I was trying to explain. However, I'd overcome the struggle and found better evidence that supported my thesis more clearly. Another struggle I faced was being open to new topics. For instance, I had wanted to do a separate topic, but because it was too similar to some of my classmates, I needed to change it.
Thank you, Huron. The next person is Ellie.
Thank you for the introduction. My name is Ellie Lin from class 702, and my focus question was, how can genetic editing affect the properties of crops used for consumption? But today, I'll be answering some questions. The first one is by Clayton from class 612. How different is 6th grade IIM from 7th grade IIM, and how can we improve our current work in a way that helps prepare us? In 7th grade, your overall IIM presentation is more rigorous, since it has to be more simple, such as writing short bullet points that you have to explain, instead of reading off of paragraphs on your slides. Also, rather than spending all your time decorating the slides, you can only pick a simple format for your slides that has to also look attractive. In addition, you have to write an essay paper that's informal. Your research is more difficult since you do not know much about your topic, and you have to write your own focus questions. Some ways you can improve your current work is by getting used to using more reliable sources, mainly in the school library research section. The second question I'll be answering is how many websites did you use? By Jason from class 612. It usually depends on your topic, but I use around 7 sources mainly. However, you may need to use many more before finding a reliable and suitable source. Sources may include journals, articles, magazines, videos, newspapers, and reliable websites. Thank you for that. Our next person answering questions is Alan Lin from class 703.
Hello and good morning, everybody. My name is Alan Lin, class of 703, and my focus question was, how can the de-extinction of woolly mammoth affect biodiversity? But today we'll be answering some questions from our friends in 613. First is from Lola Matiera. What are some tips for a presentation, visually or spoken presentation? Visually, I recommend avoiding memes as they can be distracting and shift the focus away from the content of your presentation to the meme itself. To add on, be cautious about your color palette, font size, and make your slides less wordy. Tips I will give for speech-wise is to understand it and evaluate each of your slides to stay in your own words. By practicing, you can present to your peers and be prepared with index cards to help you keep track of what you are saying throughout the presentation. Additionally, be aware of your pace and volume. Next is from Kaylee Chen. How similar is the double-line program in 7th grade compared to the 6th grade double-line program? After entering the 7th grade double IM program, I would say that there are not many similarities between the 7th and 6th grade double IM program, except for the 7 investigation method steps. But the differences are more noticeable, with the double IM program in 7th grade being more academically rigorous, being more independent, and having a sole focus on science-related topics rather than social studies. And last question is from Miranda Lee. Do you enjoy learning about the topics that you are given? Why? In the 7th grade double IM program, instead of being given a specific topic, you are provided with a theme. From there, you are expected to choose a topic that interests you, guided by the theme. For example, my topic is about the woolly mammoth, and I enjoy the topic is because I can learn more about the history of woolly mammoth and how they can act as an efficient solution to ecosystem issues. Although I enjoy the topic, I also dislike the fact that there is less information regarding the woolly mammoth compared to the topics my peers previously spoke about. But overall, the experience was enjoyable. Thank you.
Thank you for that. Now we have our last person answering questions, William from 704.
Thank you for the introduction. My name is William Oyang from class 704. My focus question was, how does genetically modified crops affect the horticultural industry? I'll be answering some questions for you guys. This one's from Luke Milton from class 604. Well, I hope you get all this information. I was not the one that presented to you today, but I can still tell you. The individuals that presented used the McKinley School Library database and searched the internet. They searched through newspapers, magazines, reliable websites, and videos that connected to their topic. The students presenting tried to decide if the source connected to their topic. A lot of the time, it did not. That did not stop them. They kept searching at home, at school, just to find a reliable source. As soon as they found the right source, they looked for key points and evidence to support their thesis. On to the next question, by Ryan Chen, 604. How do you keep calm on stage while presenting? Many students often mumble, stutter, or pause completely because of lack of confidence. This often causes anxiety and fear of presenting, which could affect future presentations and future grades. A way that has helped me keep calm on stage is to present to my friends and peers. This helps me stay comfortable while presenting. It is always better to practice with friends and peers before they can give you suggestions to work on. Another way to keep calm on stage is to memorize your slides. People often stumble and lose confidence if they cannot recall their slides. An easy solution is just to practice at home, at school, or anywhere. You could also carry index cards and use them when you're unsure, but make sure not to rely on them completely.
Now we have time for our 7th grade IIM Symposium panel, where all 10 presenters are going to go up on stage, and you will get the chance to answer any questions that you didn't get to answer earlier.
So you're going to have about another minute or so to just relax, but during that time, I want you to think of, even experiential questions are okay to ask at this point. If you have any content-specific questions, you can ask that. You might not remember who presented what, but they will be able to give each other the microphone and they'll be able to answer it for you. So just take a minute or so to think about it.
Excuse me.
Well, okay, we'll have another 15 to 20 minutes for open mic questions. Anything experiential, like I said before, and any other kind of questions regarding their presentations themselves. Anybody ready to ask questions? Start raising your hands. Okay, you know what we're going to do? I'm sorry. The first question that's out there, that's totally fine. You can ask that question. But we're going to set up the microphone here. When you have questions, start to line up in the middle. And then you can just ask questions this way. So this way, we're not running around giving the microphone out. So the mic will be open there. So start lining up for any questions you may have. If while they are answering questions, more questions arise, please quietly line up. It may be easier for you to go around the auditorium than down the center. Okay, I think that they're ready. Please state your name and class before you ask a question.
My name is Julian and I'm from class 600. If you had a choice to switch programs from IIM to, let's say, like BE, would you do it?
Personally, I would switch from IIM to BE because for me, I prefer presenting, and I feel like it's really something that I like to do in the future, and I think it's also going to help me in the future very nicely. I would.
Hi, my name is Sheila from 601. In your guys' opinion, when Ms. Leonardo's rather basic slideshow with nothing special about the research, or an entertaining slideshow targeting to entertain the audience?
Well, I would say that I'll prefer the one with more information that's boring, since it's really the physical information that matters. And if it's visually appealing, then it won't really affect the actual information that much. So I'll more so prefer the one with boring visuals, but it's not as information. You're welcome.
Hello, my name is Matthew and I'm from 604. How long did it take you to find all of this research?
It took a few months to get everything together because we had to write an essay and create a presentation. So it took a few months to get everything.
Okay.
Hi, I'm Miguel from 604, and what was the one thing that you were stressed out about the most when creating your slideshows?
Could you repeat the question, please?
What was the one thing you were stressed out about the most when creating your slideshows?
I would say memorizing all the information and the thing that stressed me out the most.
Hi, I'm Melody from A16, and what helps you to confidently improve your public speaking skills?
But personally, I think improving by practicing a lot public speech.
Hi, I'm Simone from A16. What did you find to be the most difficult
Personally, I think the hardest part about creating a presentation would be putting everything in the slides with limited words and not to make it too wordy and having enough statistics and pictures.
Hi, my name is Daniela from class 604. Would you want to go through this double IIM experience again if you have the chance?
Yes, I would, because I think I've grown a lot from this experience and have learned a lot about the topics and about myself.
Hi, my name is Paris from 816.
To answer the question that she just asked, in my case, I find IIM presentations really fun and enjoyable. Being able to present in front of my class is a really fun experience. I would definitely want to do that again.
Hi, my name is Paris from 816. I'm curious to see what made you choose your talk.
Personally, I chose my topic because I think it's something that I can connect to and many people could also connect to.
Hi, my name is Jack from class 600. Have you ever encountered any difficulties while presenting or researching, and if so, how did you overcome them?
For me personally, I think gathering the information was definitely the most difficult part because all the information I was able to gather, usually you have to put it in a time range. And in order to deal with this, I utilized the school database, the date system, trying to find more information, and by using more key terms in order to narrow down my any searches that I could actually get my hands on.
My question goes out to Jade and me of 702. Do other diseases like yellow fever go into the insides and transfer into mosquitoes the same way as malaria? And if it is different, how so?
Well, like malaria, many other mosquito-borne diseases also is transmitted through saliva, which is how malaria also spreads.
Hello, my name is Esther Zoe of class 701. How did your teacher steer you into the way of being able to be present on stage? Was it more of I want to be on the stage kind of experience, or is it like your teacher that suggested you and you're able to come on stage?
For me, it was my teacher suggesting me to present on stage.
I think it was more of Miss Leonardo saw, maybe she liked your information, she liked how you presented, so she urges you or encourages you to present.
Hi, I'm Deborah from 612, and I have a specific question for the creator of the de-extinction presentation. I want to know what exactly inspired you to do it, because I feel very inspired. I had no idea what it was until today. What really led you to doing that topic? Overall, that's an interesting topic, and among all the other topics, this one struck out to me because it was related to animals, and I did like. Yeah, it struck out to me too. Thank you.
Hi, my name is Ian of class 601, and I had a question for Safa of class 700. Were there any cons, or in other words, disadvantages of the CRISPR therapy?
Yeah, of course there were some disadvantages, like the success rate. There was a great % chance that CRISPR isn't successful, as there hasn't been many previous patients that have undergone CRISPR have a stable answer, all of their laboratory tests and results.
Yeah, okay, thank you.
I have a question for
Yes, I do. It's more effective and you don't need to worry about getting new glasses or how if it smudges up or anything like that.
I have another, my name is Ryan Scotwell from 816, and I have another question for Ingrid. Would you say that Neuralink would have like some kind of like, change, like let's say a year would change, or like a change like a Neuralink every like decade or like something like that? Like, you would have to like change like a Neuralink out of the body and push it to the
I'm sorry, I don't really understand what you're asking. Could you try rephrasing it?
So like, let's say a Neuralink could like expire, let's say, and you might have to like, get a new one, like take the old one out.
Oh, no, the actual chip doesn't expire, you just need to charge it.
Okay.
Hello, my name is Matthew from 604. What happens if the chip or the brain malfunctions and starts to spark up? Can it damage the brain?
Yes.
Or if the chip explodes, can it explode?
It hasn't been tested in many humans yet, only like 2 to 3 humans so far. So if it does happen to explode, yes, your brain will be injured and you'll probably be killed. But it has not exploded.
Or is there a chance that it could explode?
Yes.
It's still in development, so maybe.
My name is Danielle from class 604. Did you guys pick your topics or did your teacher give them to you?
Well, we got the theme of stem cells. We just had to pick a topic that's related to stem cells.
So the theme is given to you by your teacher and you have to find out the topic that's related to the stem cells?
Yes.
Personal question. Do you feel more comfortable presenting in a crowd like this or to your classmates?
Personally, I've been presenting to my classmates more comfortable because I'm used to being in front.
For me, it's a lot easier to present in front of a bunch of people that I don't know because I'm less worried about them judging me because I don't really know them.
I find, at first I found using being in front of a big crowd really nerve-wracking. I was really nervous to be presenting in front of this crowd, but I knew that my classmates and my friends were in the crowd watching me and cheering me on, so it didn't really make a difference between a classroom and in auditory.
There's from 816 again. Topic-wise, did you prefer 6th grade IIM or 7th grade IIM?
I would much prefer 6th grade IIM since it's less academically rigorous with longer deadlines, and it was more laid back, and I didn't really have to shorten information on the slides, and I could just easily read off paragraphs that I wrote on them, so I think 6th grade IIM is easier and I'll prefer it.
Hey, my name is Jaden Bloom of class 816, and my question is, do you think your 6th and 7th grade IIM teachers were a significant part of your success?
Sorry, but can you repeat the question again?
Do you think your 6th and 7th grade IIM teachers were a significant part of your your presentation. I think they were helpful.
I find both my 6th and 7th grade IIM teacher really supportive, and they're one of the reasons why I'm able to sit here and present to such a big crowd.
Thanks.
My name is Michael from A16, and I was going to ask you guys if you're excited for 8th grade IIM and getting to choose your own topic.
I'm pretty excited because I feel like I'm allowed to have I feel like I'm allowed to have more experience. I have more experience in 8th grade, and I'm able to really choose something that I'm interested in, and I think that could really help the way I present and how passionate I am.
Hello, I'm Adriana from 700, and my question goes out to Chelsea from 700. Are there any other types of animals that are currently going extinct? And if so, how can we help? Like, us as humans, how can we help prevent that?
There are various reasons why the species might go extinct. Habitat loss, human-animal conflict. So scientists can help by fixing those problems.
Okay, thank you. I have a question. You have like diabetes type 4, diabetes type 2, diabetes type 3 or 4. Which one would it be harder to learn than regular?
There are no type 3 or type 4 diabetes. However, there are hyperglycemia, which is when the body has very high glucose levels. But type 1 and type 2 are the only diabetes that can be treated by stem cells so far.
My question goes for Chelsea. And what is the cons of like de-extincting like a dinosaur? Like if you let a dinosaur come back from extinction, what are the cons of that?
So far, I have no knowledge of any company thinking of de-extincting a dinosaur.
Thank you.
Hi, I'm Ryan from class 604. Do you think today's experience will benefit you in the future?
I think my experience today would definitely benefit me in the future. This is mainly due to it's my first time presenting to such a large crowd, and I think this experience can definitely help me be able to present to a larger crowd or be more confident in the classroom presenting. So, for example, I think in the future, if I were to do a presentation, I'd be definitely more confident and more passionate in what I'm saying.
Hi, thank you.
Hello, I'm Matthew from 604. This is a question for Sinjin Lee, 704. How much millimeters of stem sperm do you need to inject into the spinal cord from S1 to T7?
That's a great question, but my research wasn't the focus of that.
Hi, my name is Daniela from class 604. Was this experience enjoyable, or was it a little hard, or it was okay?
I think this experience was enjoyable, but it was hard controlling the stress and presenting to a large crowd.
And also, what type of stress do you guys usually have if you couldn't find your information in time?
May you repeat the question?
What was some stress that you had when you couldn't find your no facts or information?
I will get nervous if I couldn't find any of the information I would need.
Okay, so my name is Alan and I have a question for you. So if somebody's born with hearing loss and they use Neuralink, will their baby also have hearing loss, or will they be normal?
Neuralink isn't something that's genetic. Hearing loss can be. So
did you find IIM in 7th grade extremely challenging and boring?
I didn't find it boring, I just found it challenging. I found it fun sometimes, but it was challenging.
To add on to that question, I believe that if you find if you have a focused question that is interesting to you, it wouldn't be as boring. Quite fun actually.
Justin Cortez. Amazing Justin What was your favorite stuff in the IIM? So the researching, big presentation,
Personally, I think the researching was my favorite part because I got to learn new things.
To add on again, I find making the slideshow and presenting is my favorite part because making my slideshow, I can see all my research getting together, and presenting, I can get to show off my artwork.
Shifting of blind teachers, if you were 6th grade last year, which one helped you more, like, help you present?
I'm sorry for interrupting, but can everyone quiet down because not everybody can hear the question.
I think Miss Leonardo was also small style due to the feedback she gave me on how to present.
What, Smith, class of 16, what generally helped you, like, decide what to do for your IIM project?
If the topic really interests you and if you enjoy researching about it, that's probably the right topic.
For J&D, how did you figure out like your topic? Like, how did you start to research for mosquitoes?
Well, I used the school database and looked in the medical database sections to see if I could find any kind of information about genetically modified mosquitoes.
I'm Sasha from 604. This is for turning skin cells into like So theoretically, like later in time, do you think it would like you would be able to like create sperm and stuff like to reproduce, or people would
I mean, in order to like to reproduce, right?
no, like you were talking about how you can create like heart from skin cells. Do you think it would be we would be able to like create like sperm from skin cells like to reproduce?
I actually do not have a lot of knowledge about that, but from what I've researched, since sperm is a type of cell, I'm pretty sure it's possible for that to happen.
Hi, my name is Danielle from class 604. I have a question. Who inspired you to like not give up on this presentation when you went through like a hard time and you couldn't find some of your facts?
For me personally, I think it was my mom who inspired me, because I feel like since she was the one who basically gave me all the courage in order to do this, so I really wanted to please her and make her feel proud.
I have to say it was probably my mom and my dad because they were very supportive to me throughout my entire IIM journey, and I wanted to come up here so that I could impress them.
I think my family was the one who encouraged me to do this because they were supportive when I couldn't get anything.
I also want to add on to that. Although my family also a big part that encouraged me, but classmates and teachers also really helped at the same time. Thank you so much.
604. This is for Jade and me. I think it's a bad idea to like kill some of the like female mosquitoes, because like it could like, first of all, they're like in the lower food chain, like they're barely doing anything, and like only some mosquitoes, like 5% of the mosquitoes can like have the diseases. And I think it's like just a bad idea to just like do that.
Well, here's the thing. Since it's invasive, so female mosquitoes or onychak mosquito that actually spread the disease, and also it's an invasive species, so you don't have to remove it from the ecosystem. So you want to remove it from the ecosystem for other wildlife to thrive, since it disrupts the very delicately crafted ecosystem. So by removing the invasive species, you're actually helping the ecosystem thrive better.
But mostly other invasive mosquitoes, they're like, they barely even do nothing. They're like, they only just do.
Can everyone please quiet down?
Paris from 816. How do you guys like plan to like use like your knowledge on stem cells in the future career?
For me, I do not plan on using the knowledge that I have as for now for a future career. I genuinely wanted to research about my topic because I was interested and I wanted to know about them, yes.
Hi, my name is Henson Zhang from 613, and my question is, why don't people in 7th grade IIM decorate their slides?
Well, they do, but it's not as noticeable because they don't want people to be distracted from what they're saying in the slides.
Hello, this is Simon from class 612. Do you plan on using IIM like in your future career or anything?
IIM is a research-based class, and it also focuses on presentation. So in the future, if I were to have a job, I think IIM really does come in handy because it focuses on research, and I would have to research, and I will have to present my information to my coworkers and my manager, etc.
What was your favorite part about doing this?
My favorite part about doing this is being able to boost my confidence and present in front of larger crowds than I usually do.
I'm Vivian from class 703. Catherine, your topic was very interesting. I just want to know what made you choose that topic all the way to stem cells.
Microgravity, my topic, stem cells and microgravity, is unique and different from other people's topics, so that's what really made me interested in it.
Hi, I'm Mr. Westberg, some of your science teachers. Out of curiosity, how many of you feel as if you might pursue science research in the future? Did this inspire you to do so?
Wow, this kind of inspired me a bit to go towards research because learning new things.
Thank you.
your attention to all of our presenters and Q&A speakers. This project took a lot of time and effort, and we're so happy that all of you were here to be able to see it, and
Our presenters have worked really hard from the beginning of the year, researching their topics about stem cells and creating their presentations to be able to share with you. But once again, thank you for coming and watching our presentations. We hope to see you next time.
They did amazing, and it's inspirational because they're 12-year-olds, right? 11, 12, 13-year-olds doing really in-depth scientific investigation. And it's hard. It's a lot of work, and I'm super proud of every one of them. I'm also very proud of all the students this year. They worked really hard. Just because these 10 students were up here presenting their findings doesn't mean that the 5 Q&A people or my 2 MCs or the other kids in the class didn't do an amazing job too, because they really did an amazing job. So I'd like to give them a round of applause as well. So I'm also really happy that it's in here. Students did, they were inspired. What they did is very inspiring. I'm very inspired by them every day. Again, I'm super proud of them. They did an amazing job. What we're going to do now is we're going to dismiss classes with their teachers by class to whatever classroom they're supposed to be in for the period.